everybody, so today's video is going to be focusing on another macromolecule, and that is proteins. So proteins are a polymer or a large molecule that have many subunits, and those subunits are amino acids. And they're basically folded into three-dimensional structures that specify the function. Protein comes from the Greek word proteos, which means first place, and proteins are the most diverse molecules in living systems in terms of structure and function. They make up 50% or more of the dry mass of our cells, and we humans contain tens of hundreds of thousands of proteins. And these proteins are made up of these amino acids, like I said, and usually these chains are made up of 200 to 300 amino acids, with the longest chains containing 34,000 amino acids. So proteins serve several functions. They serve structural functions by providing much of the supportive framework, like proteins like keratin, collagen, which is found in our hair, skin, nails, as well as tissue. They also serve the function of storage, serving as biological reserves that are used by organisms. So casein, which is a milk protein. They can also serve as transport or carriers, transporting materials. So the best example here would be hemoglobin delivering oxygen to our muscles and other tissues. Proteins also are part of hormones and signaling, delivering and carrying messages like insulin and glucagon, both proteins that regulate blood sugar levels. They also serve a function in the membrane, interacting with or being a part of biological membranes as receptors, antigens, or just part of membrane transport. Proteins are also vital for movement and motility, allowing for movement and the ability to move, like muscle contraction, which involves proteins like actin and myosin. Proteins also are part of a defensive function, such as in antibodies, fighting off infections in the human body. And finally, they also serve for metabolism, making biological or biochemical reactions possible and speeding up the rate of chemical reactions. And this is through proteins like enzymes, which are catalysts in a reaction. And in subsequent videos, we'll see the importance behind enzymes. Proteins are a polymer that are composed of amino acid monomers. And here is the basic structure of an amino acid. It consists of a central carbon that's going to be attached to an amino group, an NH2 group, and a carboxyl group, a COOH group, and a hydrogen atom, as well as a variable side group, also called an R group, that gives each amino acid its distinct characteristics. So you can see that here we have the amino and carboxyl group and the R group attached to the central carbon, and obviously that H right over there. And so there's 20 different types of amino acid side groups that can attach onto here, 20 different R groups that range from just being another hydrogen atom over here or complex carbon chains and rings. So here are just some of the side chains and R groups. So here are all the different types of R groups. And so if you were to attach these to the basic amino acid, each of them to this R group side of this basic amino acid, you would get 20 different amino acids in total. 8 out of the 20 of those amino acids are essential for humans because we can't synthesize those by ourselves, so we need those from our diet. And again, amino acids just differ based on their side groups that give them their different properties. So we can break down those side groups into different categories or groups having different types of properties, being negatively charged and acidic to positively charged and basic, or being polar amino acids or non-polar amino acids, as well as uncharged amino acids and aromatic ones. So all of them have different sorts of properties. So polypeptides are just going to be a collection of different types of amino acids, and peptide bonds are just going to link many amino acids into chains of subunits that make proteins. A peptide bond is basically just a covalent bond that's formed by the dehydration synthesis reaction between the NH2 group on one amino acid and the COOH group on the second amino acid. And what happens is when that dehydration reaction occurs, one end of the amino acid chain is going to have an NH2 group, and that's called the N-terminal end or the amino end. And the other end of the amino acid chain is going to have that COOH group, and that end is going to be called the C-terminal end or the carboxyl end. And in cells, only amino acids can be added to this C-terminal end of the growing peptide strand. So this bond that's formed between the two amino acids is known as the peptide bond. Now, the chain or polymer of amino acids that is formed by sequential peptide bonds is called a peptide. And peptides that are containing 50 or more amino acids in length are known as polypeptides. And a protein is just going to be one or more polypeptides that are folded into a precise 3D shape. And only after this folding does occur is a protein able to function. So proteins have up to 4 levels of structure, and with each level, this imparts different characteristics and degrees of complexity to the overall protein. So those levels of structure are quite simple: primary, secondary, tertiary, and quaternary, and all proteins are going to have primary to tertiary structure, whereas only some of them are also going to have this quaternary structure. So first, looking at primary structure is just the unique linear sequence of proteins' amino acids in each of the polypeptide chain, and that's determined by our DNA code. Now, changing even just a single amino acid in the primary structure will alter the overall structure of the protein to some degree, altering or destroying the biological function of the protein. So here we can see that linear sequence of each of these small amino acids, and we can see that C-terminal end and the N-terminal end, or the carboxyl end and amino end. The next level of structure is the secondary structure. So most polypeptides have portions that repeatedly coil or fold into patterns, and they contribute to the overall shape of a protein. And this structure is basically the result of hydrogen bonding that occurs between different parts of the same amino acid backbone, or just these interactions occurring between those side groups. So the most common secondary structure is alpha helix and beta-pleated sheets. So first, looking at alpha helixes, this is a delicate coil held together by hydrogen bonds between every 4th amino acid. So you can see that hydrogen bond occurring between the amino acids, and this is usually found in filamentous proteins and transport membrane proteins that provide the necessary structure for their function. The other secondary structure is beta-pleated sheets, which forms by a side-by-side alignment of amino acid chains. And this has a very important role in the strength of silk. So from the side-by-side comparison, we can see that alpha helix is just this coil, and that beta-pleated sheet is that spiral, or that folding like a fan. The next level of structure is the tertiary structure, and this is the overall 3D shape of a protein due to a range of bonding interactions among the amino acid R groups. So intermolecular reactions of the R groups determine the 3D shape of the polypeptide chain, and here are just some of the intermolecular reactions. We can have ionic interactions or bonds between positive and negative side chains attracting. We can have hydrogen bonding occurring between amino acids at different ends. We can have hydrophobic interactions, which are polar groups facing towards the outside of the water, and then the non-polar groups clustering together as a result of water. And we can also have intermolecular reactions like disulfide bridges, which are basically just covalent bonds between two sulfurs. And this is a very strong bond that holds 2 parts of the polypeptide strand together, stabilizing its shape. So here we can see that tertiary structure taking place in this polypeptide. We can see those different intermolecular reactions occurring throughout the tertiary structure. So the tertiary structure allows for the critical function of proteins, and extreme conditions, which we're going to see in a bit, can unfold the protein, causing it to lose both its structure and its function. The last level of structure is the quaternary structure, again only in some types of proteins, not all, and it's composed of 2 or more polypeptides that come together and form the final functional proteins. Now the same bonds and forces that hold a single polypeptide chain in the tertiary structure is what's also going to hold the multiple polypeptide chains together. So the best example here is the hemoglobin molecule, which contains 4 different polypeptides containing more than 140 amino acids in total. Another example would be collagen, that's made up of 3 polypeptide chains, but here in hemoglobin we can see those 4 polypeptide chains, the beta chain, the alpha chain, and it also contains a special heme group which contains iron. And so here is that structure of the heme group. So in the quaternary structure, not only are different polypeptides coming together, but we can have the addition of these alternate components like this heme group in the protein. So the overall shape of a protein can either be fibrous or globular. So fibrous proteins look like this and they provide strength. So they're long fibers. So examples of this would include collagen. We can also have globular proteins, which are going to have a spherical structure, as in hemoglobin, and this is very important for transport. So mutations are the change or the order of amino acids that's going to cause a change of the shape of the protein. So changing the amino acid sequence causes a change in the shape of the protein, which alters its functions. And some alterations can be lethal and others just change the function. So the best example would be to look at the condition known as sickle cell anemia. So here we can see a healthy person's normal red blood cell, but when you have sickle cell anemia, your red blood cell looks like a sickle. And basically, this is just a result of a small alteration in the amino acid sequence where an adenine is added instead of a glucoside. This can even be lethal because, first of all, it does not contain or it cannot carry as much oxygen, as well as it causes the cells to stick together, blocking blood flow in the arteries and the veins. Proteins can also undergo a process of denaturation, which is basically the loss of the native conformation, a change of shape, or the loss of the original structure of the protein. And this makes a protein biologically inactive and not serve its original function anymore. So this is the result of breaking interactions between different side chains, and a denaturation can either be reversible or it could be irreversible, as in the damage cannot be replaced. So different types of things that can cause denaturation are changes in pH, which change electrostatic interactions, changes in temperature, so like high fevers can be very toxic, also known as heat shock. So there's this external effect that's going to cause this internal damage, as well as changes in salt concentrations can lead to denaturation. So now we're going to look at a very special protein that helps specialize newly formed polypeptides when they to fold into their proper structure, and that is chaperonins. They have a hollow cylindrical shape, something like this, in which a newly synthesized polypeptide or protein will fit inside of, and it's refolded and fixed, and the protein with the special or proper function and structure will be released. So here's that correctly folded protein. And the inner wall of the cylinder is going to be lined with hydrophobic amino acids that's going to allow for this folding to take place. Another phenomenon that can occur between proteins that are misfolded is protein aggregation. So basically, misfolded proteins will aggregate or clump together and accumulate, and these protein aggregates are correlated to many different diseases like Huntington's disease, mad cow disease, also known as prion disease, and Alzheimer's disease. So not only is the amino acid sequence going to cause a dysfunctional protein with a different structure and no longer serving the original function, but proteins being misfolded can lead to clumps occurring, which lead to many different types of diseases.